Hey, welcome back for another biology video. We are going to continue our discussion on cell structure by now talking about the functions and roles that specific organelles that you tend to find in eukaryotic cells have. And one thing to kind of keep in mind as we go through this discussion is we're mostly going to focus on organelles that all eukaryotic cells tend to have. But we are going to specifically mention some organelles that you only find in animal cells, and maybe some organelles you only find in plant cells. So I'll try to draw your attention to that when we get there. So we're first going to start with the nucleus. So this is the organelle that is typically used as the best example of one of the organellar structures that prokaryotic cells do not have. So we'll just kind of go through it here. The nucleus's main function is that it stores and houses the genetic material, of course, that is DNA, and it's also going to serve the site for the transcription process. This is the process of the cell making specific copies of the genes that are found on DNA and turning them into a copy which we call messenger RNA, which we have talked about before. So a couple other things will happen here as well. A specific location inside of the nucleus, which is a little hard, which you can clearly see here. It's much darker than the rest of the surrounding nucleus. This is called the nucleolus. This is going to be the hub for ribosomal RNA synthesis, so somewhat similar to the transcription we see before, except this RNA that we're making is going to serve a different role than the messenger RNA does. Now, as far as the nucleus itself goes, if you for now ignore all of the DNA that is stored inside in the form of chromatin, the nucleus itself is surrounded by a set of double membranes. This is called the nuclear envelope. So you can see down here we have an inner membrane and we have an outer membrane. So this is a double membrane structure, but you'll notice here that the nuclear envelope kind of has little pores in it, kind of like Swiss cheese. So these nuclear pores are going to place a size restriction on any materials that are imported into the nucleus or exported out of the nucleus. So for that reason, if you want to move a molecule or something else from the inside of the nucleus out to the cytoplasm or vice versa, that something that you're trying to move in and out has to be smaller than the width or diameter of the nuclear pore. And then also the nuclear envelope makes distinct the cytoplasm, which is the area outside the nuclear envelope, but within the bounds of the cell membrane, and then the nucleoplasm, which is what we kind of call the area inside of the nucleus. And as we've discussed before, the area of the nucleoplasm is going to mostly be taken up by chromatin, condensed chromatin. Okay, so speaking of DNA, chromatin, and chromosomes, so we'll kind of go into more detail here on that if you kind of found what I said on the previous slide to be a little bit vague. So the idea here is that most eukaryotic cells carry so much DNA, there's so much DNA that you have to fit inside of a cell that it's difficult to imagine it all fitting inside the nucleus, let alone kind of fitting inside the whole cell itself. So if you kind of think about the actual volume of DNA that has to be stored inside of a cell, it's a lot, is basically what we're trying to say here. So how can we actually fit all of the DNA inside of the nucleus or inside of the cell itself? So the way we're going to do this is we have to compact and condense the DNA into something that has a really high amount of density, but something that takes up a lot less space. So the way that we do this is that we take individual nucleic acid molecules, so that's a whole strand of DNA, and we condense it into what is called chromatin, I mentioned on the previous slide, by sequentially wrapping it around these little spool-shaped proteins, which are called histones. You can actually see this process down here. A single DNA molecule that is just left to its own devices is not going to have much density to it, but it's going to take up a lot of volume. So if we wrap it around these little spools, like taking thread and wrapping it around a spool, we can fit a lot of thread, a lot of DNA, in a very small volume. So by doing this, we can create very dense structures, which are called chromosomes, by condensing DNA in this way. And because these chromosomes have a lot of density but don't take up a lot of volume, we can successfully store all of the DNA in a very small space like the nucleus. Now, the actual appearance of chromosomes that you see here and over here on the right is actually something that you will not see all the time. You will only see individual chromosomes, individual pieces of DNA that the cell stores in the nucleus. You will only see these structures when a cell is actively dividing, when it goes through what we call the cell division process. And that is something that we will focus on in a future chapter, so don't worry too much about that for right now. But just to kind of give you some perspective, human cells have 23 different kinds of chromosomes, and most human cells store 2 copies of each chromosome. So for any given human cell, that cell has to store 46 different chromosomes and fit them all in the nucleus. So we have to have a way of condensing the DNA so that it's all going to fit inside of the nucleus. Okay, now next let's start talking about ribosomes. This is another one that we've kind of mentioned before. So ribosomes are basically the machines that cells have that are going to interpret the messenger RNA instructions on how to make a protein and then actually stick amino acids together and form peptide bonds to make the protein itself. A ribosome is assembled out of a mixture of ribosomal RNA and protein, so it's kind of a hybrid machine made of 1 part rRNA and 1 part protein. So once the messenger RNA gets exported out of the nucleus, the ribosomes are going to attach to the messenger RNA, interpret the instructions, and then form the peptide bonds between the amino acids to start building a protein according to what the instructions say the protein should look like. Now, we're not going to focus too much on this process of what we call translation for right now. There will be a specific chapter that covers that later on, but for now, I would like you to be aware of what the general process looks like. You start with the DNA, you make a messenger RNA copy of the instructions on the DNA. The ribosome reads the messenger RNA and puts the amino acids together. That is the gist of what you should understand for right now. Okay, let's move on to a completely different kind of organelle. These are called mitochondria. You've more than likely heard of these before. Everyone knows them as these are the powerhouse of the cell, even if people don't really know what that ultimately means. So, a couple of important things to understand about mitochondria. If you look at one of these kind of textbook images of what an animal cell looks like, you see there's only 2 or 3 mitochondria listed here, and that is not at all accurate. Most cells are usually going to have hundreds, if not thousands, of individual mitochondria per cell. Just like with the nucleus, this is a double-membraned organelle in the sense that it has an outer membrane and an inner membrane. So this is going to leave us with 2 different cavities within the mitochondria itself. The innermost cavity is called the matrix, okay? So the matrix here is going to be where a lot of the enzymes that are necessary for cellular respiration are stored. And then you have what is called an intermembrane space, which is the space in between the inner and outer membrane, and a couple of important things will happen there as well, and we will learn all about those when we get to chapter 7, I think it is. And then one other thing that is important about mitochondria, even if it sometimes flies under the radar, these are more than just energy factories. They are more than just the powerhouse of the cell. They do lots of other interesting things as well. In fact, one of the things they participate in is helping a cell move along through programmed cell death, which is called apoptosis. It sounds like a bad thing, but sometimes it is absolutely necessary for the health of an organism. So the main thing that mitochondria are doing is they facilitate cellular respiration, and you can see the overall reaction scheme for what cellular respiration looks like. You start with a molecule of glucose, that's C6H12O6. You combust it with 6 equivalents of oxygen, and that will yield 6 equivalents of carbon dioxide, 6 equivalents of water. The molecule of glucose stored a lot of energy, so in breaking those chemical bonds, you also release quite a lot of energy, and that energy, the cell is going to package inside of this molecule here that we have learned about before, called ATP, okay? So the main thing I want you to understand for right now is that the reason why glucose is listed here, it's not because glucose is the only type of molecule that can participate in cellular respiration, it's just the most common one. So any nutrient molecules like glucose, other types of sugars, fatty acids, we can put those through cellular respiration, and that is how our cells obtain energy from the food that we eat. Through the digestion process, we break down complex carbohydrates, proteins, fats, and things of that nature into their monomeric forms, and in doing so, we can then absorb those monomers into the bloodstream and then deliver them to cells all throughout the body. And as long as those cells have mitochondria, and as long as they can get their hands on oxygen, then those cells can extract a lot of the energy out of these glucose molecules or fatty acid molecules or whatever. So like I said, a portion of the released energy, it's not 100% efficient, so we are going to find out when we get to chapter 6 that we are definitely going to lose some potential energy in the form of heat, but some of the released energy can be packaged into the high energy bonds of adenosine triphosphate. That is the energy currency that our cells use. So the idea here is that since these mitochondria are like little energy factories, little ATP producing factories, if a cell or a tissue or an organ inside the body of an organism requires a lot of energy, we would expect those cells to contain a lot of mitochondria. So how many mitochondria a cell contains is going to be reflective of the energy needs of that cell. And then one other thing to kind of take note of, and this is something that we talked about in a previous chapter, the fact that carbon dioxide is released as a byproduct of cellular respiration, that is cause for concern because we saw before that carbon dioxide, by becoming carbonic acid once it hits an aqueous medium, it can cause the surroundings to become more acidic. So the organism that performs cellular respiration is going to have some sort of way of dealing with that, of making sure that the pH does not change too terribly much. Okay, moving on to a somewhat related organelle. These are chloroplasts. Again, these are double membrane structures. They contain an outer membrane and an inner membrane, but these are organelles that animal cells do not have, because they serve as the site of cellular respiration, meaning that these types of organelles are only going to be found in the cells of eukaryotic photosynthesizers, that is, plant cells and algae cells. So we talked before about what kinds of organisms are photosynthesizers, and we said that is plants, algae, those are eukaryotic protists, and then cyanobacteria. But cyanobacteria are prokaryotes, so therefore they do not contain organelles. So we'll talk a little bit about how they perform photosynthesis here in just a little bit. So like we said, the chloroplast serves as the site of photosynthesis in the cell. So if a cell is going to be capable of performing photosynthesis, this is the organelle it has to have possession of. So photosynthesis, we are going to learn all about when we get to chapter 8. This involves the capture of energy from the sun, the consumption of water and carbon dioxide, and in doing so, we are going to produce oxygen and then stored up nutrients like sugars, carbohydrates, and things like that. Essentially, this is the exact mirror opposite of cellular respiration. All the things we are required to start with here, energy, carbon dioxide, and water, are the things that are byproducts of cellular respiration. Now, if you're wondering why this whole structure is green here, that is because the chloroplast contains a lot of the photosynthetic pigment, which is called chlorophyll. In a later chapter, we will cover why chlorophyll appears green and therefore why plants appear green to us. But for now, I'll just kind of tease. It's the fact that this pigment absorbs certain wavelengths of light while reflecting others. The green wavelengths of light are the ones that are reflected at our eyes, which is why plants appear green to us. Okay, so what we're going to see here is that mitochondria and chloroplasts ultimately work together. So the idea is that photosynthesizers are a type of organism called an autotroph. They make and consume their own food. The idea is that a plant, for example, will absorb energy from the sun, perform photosynthesis to make carbohydrates that it then stores up inside of its own cells, and then the plant cell will also have mitochondria that makes these plant cells capable of consuming the food that they made via photosynthesis. So a photosynthesizer or an autotroph will not only make their own food, but they will eat their own food as well. They do not need to consume any other organisms or eat the byproducts of other organisms. They make their own food. So food made during photosynthesis is, like I said, consumed through cellular respiration to produce ATP for the cell's usage. This is a common misconception. Plant cells have both chloroplasts and mitochondria. It is not an either-or proposition. Cells do not have either mitochondria or chloroplasts. Plant cells have both mitochondria and chloroplasts. The chloroplasts allow them to make the food. The mitochondria allow them to eat the food and get the energy out of it. So what about us? What about us organisms like humans that are not photosynthesizers? Well, we only have mitochondria. We do not have chloroplasts. We can consume food and get the energy out of it, but we can't make the food through photosynthesis. So we are called heterotrophic organisms. We can consume food, but we can't make our own food, so we have to obtain our food by eating other organisms or eating the byproducts of photosynthesis, which come from plants. So that's fruits and vegetables. Okay, so let's tie this discussion together by talking about endosymbiotic theory. This is basically a very kind of philosophical discussion on where did mitochondria come from, where did chloroplasts come from, and heck, where did the nucleus come from? So if you'll recall back to chapter 1, we discussed the idea that all organisms on Earth have a common ancestor. So if that's true, at one point in ancient history, there was no distinction between prokaryotes and eukaryotes. There was only one kind of cell. So how did we get a divide between prokaryotes, which do not have organelles, and eukaryotes, which do? So where did we as eukaryotes get our organelles from? So endosymbiotic theory is an idea that potentially explains this. So the idea is that mitochondria, chloroplasts, and maybe even the nucleus, these individual organelles, so forget about the whole cell, we're just talking about the organelles, they actually have a lot in common with prokaryotic organisms. Both the mitochondria and the chloroplasts are about the same size as a prokaryotic organism. They've got their own DNA and RNA, they've got their own ribosomes. So there's a lot of evidence to suggest that at some point in history, mitochondria and chloroplasts were actually independently functioning prokaryotic organisms. And the idea here is that they were engulfed by a much larger cell, and basically they just decided to start living there. So this became a symbiotic relationship between the cell that engulfed the mitochondria and chloroplasts and those two organelles themselves. So if you're not exactly sure what that term means, symbiosis is a beneficial relationship between two different organisms. Basically, it's a kind of relationship where both organisms benefit from the relationship itself. So it's kind of like an I scratch your back, you scratch my back kind of relationship. So they help each other divide by living together, growing together, and helping each other get what they need. So the idea, therefore, is that the cell here, this modern eukaryote, which at some point in history engulfed a mitochondrion or engulfed a chloroplast, because the cell did that, it now becomes capable of cellular respiration and photosynthesis. It is in a much better place than it was without the mitochondrion or without the chloroplast. And from those two organelles' perspective, they are in a better position too because they don't have to fend for themselves anymore. Basically, any nutrients that the larger eukaryotic cell gets their hands on now becomes fodder for them to use, so they don't have to worry about getting their food anymore. Okay, so a couple more organelles for us to discuss. First, we'll just kind of briefly mention peroxisomes. These are small organelles that basically do a couple of different things, namely oxidize fatty acids. So if you're not using sugars for your food, you're probably using fatty acids, and a lot of the initial oxidation reactions that kind of help to break those fatty acids down occur in the peroxisomes. But one of the major things I want to discuss here is the neutralization of what is called reactive oxygen species, or ROS. So what is this? Well, if we think back to cellular respiration consumes oxygen, which organisms get from the air. Well, that can sometimes be a little bit of a messy process in reducing oxygen into something that's not completely dangerous. So sometimes the cell doesn't handle this very well, and the cell will produce a harmful molecule called superoxide. So this is a very highly reactive molecule. If it's allowed to run rampant inside the cell, it can damage the DNA, damage proteins, damage cell membranes, so it can cause a lot of damage to cells. Peroxisomes contain two enzymes that help to neutralize superoxide molecules when they get created. These two enzymes are called superoxide dismutase and catalase. We may talk about these two enzymes in the future, but for now, it's just enough to kind of know that they exist. Okay, our next organelle is the lysosome. So if you had to kind of come up with a role for lysosomes that kind of matches the role of maybe like a part of a town or a city, this is kind of like the garbage dump or the recycling plant for the cell. So these are small organelles that contain a number of different hydrolytic enzymes. That word hydrolytic should seem familiar to you. Basically, it means that this is an enzyme that facilitates and catalyzes hydrolysis reactions. Remember, these are the reactions that break down polymers into smaller monomeric units by breaking the covalent bonds that join those monomers together. An interesting thing about lysosomes is that in stark contrast to the rest of the cell, they like a very acidic environment. So if you were to actually measure the pH inside of a lysosome, it's going to be pretty acidic, somewhere around a pH of 4, as compared to a pH of 7 pretty much everywhere else. So like we said, these enzymes catalyze the hydrolysis of macromolecules, so proteins, carbohydrates, lipids, nucleic acids. If you want to break them down and destroy them and reconstitute and recycle the individual monomers, this is the place to do it. So oftentimes what you'll see inside of a cell is that transport vesicles that contain molecules that need to be digested like this, or maybe even much larger materials like whole organelles. These vesicles can fuse with the lysosome and introduce the molecules or materials that need to be destroyed, introduce them to those digestive enzymes, and then the breakdown can proceed from there. In this electron micrograph, you can see here, you can actually see a whole mitochondrion that has been engulfed by a lysosome. The idea here is that this mitochondrion and maybe this peroxisome are old and maybe no longer functioning the way that they should, so they become engulfed by a lysosome so that the lysosome can destroy them and recycle the individual materials to make new mitochondria or to make new peroxisomes. Okay, and then the last, this isn't really an organelle per se because it's not a membrane-bound structure, but one structure that you find inside of eukaryotic cells that I do want to mention are called proteasomes. So proteasomes are just big protein complexes that have different types of proteolytic activities. It's basically just a big conglomeration of enzymes that break down proteins via hydrolysis. So the idea here, what makes this different from the lysosome, is that the lysosome is just kind of a general garbage dump. Anything you want to destroy, just throw it in there. Now, proteins are a little bit different because oftentimes proteins need to be specifically degraded or specifically destroyed. You don't want to destroy every protein in the cell, you just want to destroy this one right here or this specific one right here. So that is what the proteasome is for. So if you have a very specific protein inside of the cell, and keep in mind there's tens of thousands of different kinds of proteins inside the cell at any given time, if there's just one particular one that needs to be destroyed, you can tag that protein with a little small protein called ubiquitin. It's kind of like putting a little stamp on it, and then that is going to be recognized by the proteasome. That ubiquitin-tagged protein will be delivered here. It kind of looks like a garbage can that has two different flaps to it. The protein will be threaded through the cylinder here, and the enzymes that are inside the proteasome will start breaking peptide bonds and regenerate individual amino acids that can then be used to build new proteins. It's kind of like a recycling program. Okay, that is going to do it for our overview of organelle function. We will discuss more of these organelles in future videos, so what we've talked about here is just a little bit of a taste. So thank you for your attention, and in the next video we are going to start talking more about the nucleus, DNA, other organelles that we have not mentioned yet, like the endoplasmic reticulum, organelles that make up what is called the endomembrane system, which is important for protein synthesis. So we're going to continue some of our discussions from this video in a little bit more detail. So thanks again, and I will see you next time.